Hello, in this lecture we shall talk about the vegetative reproduction in bryophytes. So what are bryophytes? Bryophytes are a group of plants which occupy the aquatic as well as the moist habitat, and hence they're called as amphibians of the plant kingdom. These are primitive group of plants. Bryophytes can be broadly classified into three classes, namely Hepaticopsida, Anthoceratopsida, and Bryopsida. They're commonly called as liverworts, hornworts, and mosses. Liverworts, as they appear liver-like or lobe-like in appearance, while Anthoceratopsida or hornworts, they appear horn-like, that is nothing but the sporophyte which appears on the surface of the thallus and horn-like in appearance. Bryopsida, or otherwise called as mosses, they have a leafy gametophore. So let's talk about vegetative reproduction in bryophytes. There are several methods of vegetative reproduction. The first, by the death and decay of the older parts of the thallus, or otherwise called fragmentation. This is the most common method of reproduction seen in bryophytes. For example, in Riccia marcantia. The progressive death and decay of the older parts of the thallus reaches to the dichotomy. The lobes then get detached, and these lobes can individually grow into new thallus. Or they can accidentally break up into small fragments, and each of the fragment can develop into a new thallus. Second, by tuber formation. Tubers are basically seen at the end of the growing season, particularly at the apices of the thallus or along the margin of the thallus. They formed during unfavorable conditions and store starch and oil droplets. On the onset of the favorable condition, the tubers produce new thallus. Example, it is seen in Riccia, Anthoceros, Frossimronia, etc. The next method of vegetative reproduction is by gemma formation. Gemmae are small, multicellular green reproductive structures which are present on the surface of the thallus. They are seen particularly on the dorsal surface along the midrib region. At maturity, they get detached from the parent plant, and after falling onto a suitable substratum, the gemma gives rise to new individuals. These are seen in Marcantia. The next method is by which adventitious branches. Several apices of Riccia produce adventitious branches from the ventral surface of the gametophyte. These branches, on being detached, may result in the formation of new gametophytes. Such adventitious branches may have also been recorded from other bryophytes. Example, Marcantia, Anthoceros. By innovation. In certain mosses like sphagnum, one of the branches in the apical cluster, instead of forming drooping branches or divergent branches, develop more vigorously than the others and continues the growth upwards. This is called innovation. Due to progressive death and decay of the parent plant, these innovations become separated from the parent plant and establish themselves as individual plants. By bulbil formation. Bulbils are small resting buds which develop along the rhizoids. Bulbils are devoid of chlorophyll but full of starch. On germination, the bulbils produce a protonema-like structure which bears the leafy gametophores. By primary protonema. In mosses like Funaria, the primary protonema is nothing but a small filament which results by the germination of the spores. The protonema may break into small pieces, and each such fragment is capable of growing into a new individual by the production of buds on them. By secondary protonema. The protonema formed by other methods then the germination of the spores is called as secondary protonema. The secondary protonema may arise or develop from the leaf, stem, rhizome, or from any injured part of the plant. From this, the new gametophores or lateral buds arise in the same manner as that of the primary protonema, seen in Funaria and Sphagnum. The next method of vegetative reproduction is by cladia formation. Cladia are small or broad detachable branches, which are of two types. They may arise from the leaf, called as leaf cladia, example Prolania, or they may arise from the stem, which is called as stem cladia, and occupy the same position as sexual branches, example Bryopteris. The next method is by persistent episis. Due to prolonged summer or towards the end of growing season, the whole thallus in some species dries and gets destroyed, except the growing point. Later, it grows deep into the soil and becomes thick. Under favorable conditions, it develops into a new thallus. It is a method of perennation than multiplication. Thank you.